And, you know, I think it's time to do this. Our neuropathology group here at Columbia spent a long time and looked at a very large number of autopsy brains from our COVID patients. And I just wanted to discuss with you what we find and ask some questions about whether or not what we find can possibly guide us in understanding neuropsychiatric symptoms in people who survive. I can't promise I'll have any answers, but at least it'll give us something to think about. So we all know that post-COVID syndromes include a lot of neuropsychiatric symptoms, and this is just a long list of them. Cognitive disturbances, anxiety, headaches, sleep disturbances, autonomic dysfunction, persistent anosmia, and all of these things. Then, of course, there are the consequences of the acute infection and hospitalization. And finally, a lot of patients show an exacerbation of the existing comorbidities. And one or more of these things has been reported in anywhere from a few percent to as much as 35 or 40% of COVID survivors. So it's pretty common. We and other medical centers have investigated the neuropathology. I've shown you, because we're at Columbia, I've shown you our two Columbia papers. This one came out last year in Brain. But there are a number of other of our neuropathology and molecular colleagues who have also studied COVID brains and come up with some of the same conclusions. Okay, so what is it that we find? And I'll go through these in some detail in a little while, but almost all these patients show diffuse hypoxic changes in their brains. And that's probably largely due to the severe pulmonary disease they get. Some of them have infarcts, large infarctions, but usually small and hemorrhagic. And that suggested to us that they're thrombotic or thromboembolic events. The blood clots block the small vessels and then break up, and the blood flows into a damaged area and causes a hemorrhage. Inflammation, and it's an interesting inflammation because there's a lot of microglial activation with neuronophagia, which is a phagocytosis and killing of neurons. Interestingly, there is relatively little lymphocyte accumulation. T cells, CD8 and CD4 T cells, this contrasts dramatically with most viral encephalopathies, encephalitides. One of our patients had a herpes, HSV1 encephalitis, and that brain looked completely different. There's an increased amount of inflammatory molecules. We have evidence for disturbances in neurogenesis in hippocampus, as I'll show you some new data on beta-amyloid deposition. Astrocytes are reactive. Others have found Alzheimer-like changes, in particular tau hyperphosphorylation. And there are rare other things that people have described. And I'll illustrate some of these, but first I want to try to clarify something, and that is there any virus in the brain? And I have to tell you, my opinion, it's a controversial subject still, but my opinion from our study and many others that there is little or no detectable virus in any of these brains. And the reasons I think that are that rt-PCR levels are very low. And since with rt-PCR, you grind up the tissue, what you find may reflect viral RNA in blood or in blood vessel cells or meninges. Secondly, in situ hybridizations that we've done are negative. Thirdly, immunostaining is generally negative. There are a few studies that purport to show some positivity in CNS cells, but frankly, I'm not completely convinced of those. There are two papers that show single nuclei RNA sequencing of brain tissues. These have not identified viral sequences. We know that virus is present in nasal and olfactory epithelia. The paper just came out from Stavros Lomvardis' lab in collaboration with a lot of us at Columbia and Ben Tenover. And they find the virus in the sustentacular cells, that is glial cells, in the olfactory epithelium, but not in neurons. Thus, the virus can't be transmitted into the olfactory bulb via neuronal axons. Klein at Washington University have looked at hamster models using intranasal infection, and they don't find virus in the brain. So for all these reasons, I am not convinced that there's virus in the central nervous system. Nevertheless, there's a lot of pathology. So here's just a couple of pieces of data. This is something that Emily Miller and Ann-Katrin Ullman did at Columbia with 26 of our COVID patients in their brains. PCR, nasopharyngeal swab, CT levels were low, which means high levels of virus. In nasal epithelium here, they were also on the low side. But if you look at brain tissues in the olfactory bulb, frontal and temporal lobe, cerebellum, medulla, with one exception here, they're extremely low. And as I said, this could represent viral RNA in blood, CSF blood vessels, rather than in brain tissue itself. So Mike Glendening and Dritan Agulu here at Columbia did a lot of in situ hybridization. These are some in situs that they did with N plus N and S region probes. They were unable to find anything. These are just two examples. Yet, as a positive control, when they did the same things in lung, there were lots of viral in situ RNAs. We also did a lot of immunostains with anti-S and anti-N antibodies, and those were all negative, and with positive controls being lung and nasal epithelium. So with that in mind, let's see what we find in the brains. The first thing is that a lot of these brains showed infarcts. And here's one example. Here's a CAT scan here and here, an infarct in one of our patients. The neuroradiology team did a post-mortem MRI on a brain slice. It shows you the same thing here and here. And then we were able to look at the slice of fixed brain tissue that corresponds to this. This is reversed. That shows you this. It's in the same place. And if you look at this under the microscope, there's a large area of necrosis, death, and then there's a large area of hemorrhage. So these are small hemorrhagic infarcts. Here's another one, here, and a hemorrhage. So as I said before, these acute hemorrhagic infarcts imply thromboembolic or thrombotic occlusions. Other people have found thrombi in brain vessels. We found a few of those. Okay, one of the things that was very dramatic was that we immunostained a lot of brain tissue with an antibody to CD68. It's a lysosomal protein that's increased in activated microglia. And we looked in many areas of the brain. And we found activated microglia in cortex, white matter. They were particularly prevalent for some reason that I'll discuss later in the lower brainstem, pons medulla, and in the cerebellar nuclei. More about this later. So here's what we see. Here's a brainstem nucleus showing neurons, and on a hematoxylin eosin, there's a cluster of cells here. These turn out to be microglial cells. And when we stain them with CD68, we see this microglial cluster. Here's another one here. Here's another one here. And these are associated with neuronophagia, that is the phagocytosis and killing of neurons by microglia. Here's a stain in the locus ceruleus, neuroadrenergic nucleus. It projects widely throughout the brain. And here's one of these neuronophagic nodules. If you look at these higher power, you see a neuron in the center degenerating and microglia surrounding this. And these are from the dentate nucleus of the cerebellum. And these are also neurons surrounded by activated microglia. Now, why do microglia kill neurons? there's something known about this. And there are a number of things that can trigger this. Hypoxia is one. Cytokines are another. And beta-amyloid, that I'll talk about in a minute, can also do this, and other things as well. So one of the interesting questions is, do neurons in the COVID-19 brain express any signals that promote their own death? And that's something we would like to look at, and I think it's possible to look at it. Interestingly, as a control for COVID, we found a number of brains from non-COVID patients who died with adult respiratory distress syndrome. That is, people who had severe hypoxia and died, but not because of COVID. And we stained them with CD68, and we found the same type of microglial activation in their brains, including the brainstem, perhaps implicating hypoxia as a contributing factor. And thus, this, in fact, many of these pathologies are, frankly, not specific for COVID-19, even though they're disastrous. Allison Song and Robin Klein at WashU did some studies on tissue that we sent them. And here's one of their studies that showed interleukin-1 beta immunostaining in the medulla of one of our patients. This is the control patient. The interleukin-1 beta is in green. The IBA1, which is a microglial marker, is in magenta. And the blue shows DAPI, which is a general marker for all cell nuclei. And what they found was that there was an increased amount of IL-1 beta, and that most of this IL-1 beta could be co-localized with microglia. They've done some other studies as well. So there seems to be inflammatory molecules that are increased in these COVID brains. Some of them may be made in the brains themselves, and others may come in from the blood and CSF. Robin's lab in hamsters and Maura Boldrini's lab at the in psychiatry here that work with human neurogenesis in the hippocampus also looked at some elements of hippocampal neurogenesis. And Robin's lab found that this is what they've done. They've stained hamster dentate gyrus, where the new neurons are being made. Again, DAPI stains all of these nuclei non-specifically, but they looked at double cortin, which is a measure of immature neurons, but not mature neurons. And Ki67, which is a cell cycle marker. And they found, usually you find Ki67 in proliferating precursors in the normal dentate. And you also find that a number of these precursors are cycling, so that they're Ki67 positive, double cortin positive. And what they find is that after an intranasal injection of COVID into the hamster, they find early on there's no change, and then by 4 days after infection, there's a marked drop-off in cycling cells. Cycling cells and cycling cells that are double cortin positive. A marked drop-off, but these recover somewhat. Maura looked at, again, the same area in the human brain, the DAPI showing the all of the nuclei, NeuN, which is a neuronal transcription factor, in green, showing the neurons, and double cortin showing immature neurons. So they express double cortin before they express NeuN. And so they looked at cells that were double cortin positive, but NeuN negative, that is, they were precursors, like this one here. And they found in the controls versus COVIDs, these are non-COVID controls, that there was an appreciable decrease in the numbers of double cortin positive cells that had not yet expressed this neuronal marker. So both of these give some indication that there may be some neuronal dysgenesis in the hippocampus in our COVID patients. Now, we found a lot of changes in the brainstem, and I hate to get into the weeds here, so you'll forgive me, but I have to get into the neuroanatomy weeds. So please just follow along if you can. The medulla, which is so important, containing cardiorespiratory centers, chemoreceptive centers, centers that regulate wakefulness, centers that regulate a lot of autonomic output. And in particular, this thing, the solitary nucleus here, receives input from the vagal nucleus and from baroreceptors, chemoreceptors, and it senses blood pH, oxygen, CO2, lung stretch receptors, and aldosterone. And so this is getting a lot of input from other organs in our COVID patients. It sends output to a number of things, including this dorsal motor nucleus of the vagus and an area called the nucleus ambiguus, which send a lot of autonomic controls out through the vagus nerve to a number of different organs. So the output of this goes to this dorsal motor nucleus of the vagus, nucleus ambiguus. So those have a lot of control over preganglionic neurons for autonomic reflexes. It also sends output to the hypothalamus, particularly the paraventricular nucleus, that activates their corticotropin releasing factors that go to the pituitary gland, releasing ACTH that goes to the adrenal medulla that activates glucocorticoids, which tends to damp down inflammatory reactions. But this also sends input to the amygdala, to the locus ceruleus and raphe nucleus. This is a big noradrenergic nucleus and a big serotonergic nucleus in the medulla and lower and pons that project all over the brain and do a number of things, but including regulate wakefulness states. Okay, so here's another one, and don't mind all of the anatomy here. I just want to focus on a couple of things. One is this. Here's the vagal nucleus, the afferents, from here in various organs into the solitary tract nucleus that I just talked about. These vagal endings can be stimulated by pro-inflammatory cytokines. And as an example, many of them have receptors to interleukin-1 beta and other inflammatory cytokines. So they are able to convey inflammatory signals from outside the brain into the medulla of the brain. And as I said before, this nucleus sends and synaptically excites a number of different areas like the dorsal motor nucleus of the vagus, nucleus ambiguus, that then send a lot of afferents or efferents out into the liver and GI tract and intestines and cardiac. These, for example, contain cardiac or cardioinhibitory preganglionic parasympathetic neurons. So all of this can affect the autonomic systems that regulate a lot of different functions of our peripheral organs. And all of this is sent into the brain and then sent back out of the brain from the medulla. Okay, now we could also see direct hypoxic effects on brainstem neurons. Remember, these people are very sick, and they really have severe pulmonary disease, and so they're hypoxic. So you can get direct hypoxic effects. And that can lead to microglial activation and neuronal killing. Also, some of these patients experience drops in blood pressure, hypotension. And the brainstem has several what we call watershed areas. That is, areas that are at the ends of arterial distributions into the middle of the brainstem. And those areas are most sensitive when blood pressure drops because they're at the ends of these arteries. And so they tend to get less blood flow. Hypoxia also activates nucleus tractus solitarius neurons that project to the paraventricular nucleus of the hypothalamus, which is involved in respiratory control. So there's a lot of stuff going on here. Again, in the brainstem. Okay. Something that we didn't expect, but we found, this is unpublished now, that we've done in collaboration with Harker Rhodes, Dan Pearl, David Premer, and Esma Karlovich. These three are at the Department found was beta-amyloid deposits in the brains of COVID autopsied patients. And this was kind of unusual because we looked at, particularly at patients who were less than 60 years old. Now, the data on, the huge data on amyloid and Alzheimer's disease tells us that it's very unusual to see any amyloid deposits in the cortex of people less than 60. Obviously, once you get higher than that, and you get up into 70s, into 90s, you start seeing more and more of that. But there's very little of this. So what we see here, this is one of our COVID patients' frontal cortex stained with a beta-amyloid stain. And you see dark staining here. This is one, here's another, another. They range from a few to a great many. If we look more closely, not only do we find these deposits that we think are the neuropil that is outside of neurons, but we also find this stuff inside neurons. And here are two pyramidal neurons. We think this is probably APP, the precursor of amyloid, because this antibody also reacts with APP. Interestingly, we find rather little or no tangles, that is phospho-tau immunostaining in these regions. So we think that these deposits are not a sign of Alzheimer's disease in these younger patients. Whether or not they could be precursors, we have no idea. Then we did two controls. As one control, we examined a lot of the brains in the Department of Defense military traumatic. And what Dan found was that the large majority of those showed no amyloid deposits at all. Only 5 of 61 showed any amyloid deposits. 1 or 2 of these we know had sustained hypoxia. And he's looking at the clinical features of these 5 to see if we can distinguish them. But in general, you don't find these amyloid deposits in this kind of control. And then as another control, we looked at people who did not have COVID. This was prior to the COVID pandemic, but who died with ARDS, adult respiratory distress syndrome, and had severe hypoxia. And here's one of them, frontal cortex, patient with ARDS, less than 60, who had tons of these amyloid distributions, and here too. And we found these in the neuropil, and we also found them in cell bodies. So this may implicate hypoxia, again, as a contributing factor, perhaps with inflammatory systemic inflammation also. Now, of note, there have been a lot of clinical studies in patients who survive ARDS. And the clinicians who take care of these folks, neurologists and pulmonologists, have noted that a number of these people experience cognitive deficits. And that was interesting. Now, there's been no pathology done on these, or very little, except for this and a few other studies. Could this be hypoxic damage to neurons? Is it amyloid accumulation? Is it inflammation? No one knows. So the bottom line here, though, is that are these deposits harmful? Well, who knows? They might not be. Supposing the brain is able to get rid of, this is soluble amyloid. Supposing the brain is able to get rid of it. The mammalian brain can get rid of small molecules out into the cerebrospinal fluid. And we do this rather efficiently. We don't know whether these are gotten rid of. There is also evidence from mouse and a couple of papers in human Alzheimer brains or models that the beta-amyloid oligomers, small molecule beta-amyloids, are very synaptically toxic. And one of the things we'd like to look at in these brains is there any evidence for synaptic toxicity? Because that might tell us something about things like cognitive states. Okay.
Jim, a couple of minutes, Jim. Yeah.
Okay, we're almost done.
Great.
Okay, what do we know so far? So these brains show a range of neuropathologies, even in the likely absence or low levels of virus. None of the pathologies appear to be specific to SARS-CoV. And if patients who survive the infections also have these changes, that's a big if, they may be plausible reasons for thinking that they could produce a number of clinical features. Now, there are limitations to these studies. First of all, they're autopsy studies. So these are the most severely ill patients. Would patients who recover develop any of these? Now, we've examined the brains of several patients now who have recovered and then died weeks to months later, and we find some of the same pathologies. But we don't know if that person actually had developed those pathologies at the time of the acute infection, or the circumstances around the death could have produced this. Hypoxia is a good example. We're investigating some of the hamster and mouse models because then you can follow these. And if they show some of the same things, we can see if the changes are reversible. And finally, a lot of the patients show comorbidities, and those may have contributed to neuropathology. So finally, this has been work of a huge number of people, so many I can't put their pictures up, at Columbia, in pathology, in neurology, neuroradiology, molecular biology, here at Presbyterian Hospital and at New York State Psychiatric Institute, at Washington University, at Mount Sinai, at Uniformed Services. And I'd like to give a particular shout out to our autopsy staff who worked under very difficult conditions, day and night, to get this material for us. And they are to be really applauded for their efforts. It was very difficult. And I'll leave you with this, which is from our brain paper, kind of a cool graphic done by Trina, who turns out to be Peter Knoll's wife. So we kept it in the family. Anyway, thank you very much. And I'll be glad to answer any few questions.
Jim, thanks very much for that. It sounds like hypoxia is a huge thing. I mean, it could be almost everything that's going on in this. So you didn't talk too much about long COVID. Do you think these are, explanations for the long COVID brain fog?
Who knows? I mean, the problem is that, as I said, we've looked at people with long COVID, and who've survived for a long time, and they show some of these same changes. But the problem is we don't know when those changes arose. And I think we may, if some of these models show some of this, like amyloid deposition, we will be able to look at those over long courses to see if the brain's able to deal with this stuff. So, I think it's something that's important to keep in mind, but it's going to be difficult. Note that these amyloid deposits, since they're soluble amyloid, are not going to show up on amyloid PET scans. There are some really interesting things that discussions of amyloid and phospho-tau in serum and cerebrospinal fluid that have been positive that may allow us again to correlate with some of these things. But at present, it's really difficult to do this.
So we have one question from Stuart Firestein, who says, is there any evidence for breakdown of the blood-brain barrier in COVID patients?
Okay, that's an interesting question, Stuart. There is. You can see if we stain some of these sections with with things like albumin or immunoglobulin, we see in some patients a ring around the blood vessels. So there may be some, certainly in the infarcts, this breakdown of the blood-brain barrier for sure, in hypoxic infarcts. But it doesn't seem to be extensive outside of infarcts. But we all know that lymphocytes and monocytes and molecules are able to get into the brain through an intact blood-brain barrier.
All right. So we've got 2 raised hands. Eric, go ahead.
I think Rodney had his hand up first, but I'll go ahead anyway. Thanks for your talk, Jim. Very informative. You touched on this, and I just want to make sure that I caught it correctly, and if not, clarify my question. You said you looked at some brains of folks that had survived COVID and then passed away for some other reason down the road. And I'm just curious, I wanted to know, those patients had reported having long COVID neurological symptoms. Is that correct?
Some of them. We need to interrogate their medical charts very, very carefully for this.
Okay. And have other people
to see the details?
Yeah, I understand.
Other people have done that. It's not been published. I know this through the neuropathology and neurology grapevines that I'm attached to, but it's really not been published much.
Yeah. Okay.
So let's wait. I think over the next 6 months to a year, more of this is going to come out.
Okay, Rodney, can you ask your question? Yeah. Great.
Yeah, I'm unmuted. Thanks. That was a really interesting talk. And my question is very similar to Eric's. And that question is, it has to do with how a person dies. Okay, so in other words, if, like you said, if somebody dies and you don't know whether it's hypoxic versus not, et cetera, whether they had hypoxia, what about just looking at people who died in car accidents, or something that was unrelated in a sense to the infection? Have you done that kind of thing? And what does the plaque look like in brains like that?
Rodney, that's a fascinating and very important question, because a lot of people die with pneumonia or other pulmonary or cardiac disease. I think the best data we have so far is from Dan Pearl's work in the Department of Defense. A lot of their patients who develop PTSD, non-injuries, die of other things. They die in motor bleed. Many of them suffer severe depression and commit suicide.
Yeah.
And he's looked at many of those brains now. And he's a neurodegenerative disease person. He's stained them all for tau and amyloid. And he finds in the large majority of these, there is no amyloid deposition in the cortex or anywhere else in these brains.